Section 5.3, the Calvin cycle. By the end of this section, you will be able to describe the Calvin cycle, define carbon fixation, and explain how photosynthesis works in the energy cycle of all living organisms. After the energy from the sun is converted and packaged into ATP and NADPH, the cell has the fuel needed to build food in the form of carbohydrate molecules. The carbohydrate molecules made will have a backbone of carbon atoms. Where does the carbon come from? The carbon atoms used to build carbohydrate molecules comes from carbon dioxide, the gas that animals exhale with each breath. The Calvin cycle is the term used for the reactions of photosynthesis that use the energy stored by the light-dependent reactions to form glucose and other carbohydrate molecules. The interworkings of the Calvin cycle. In plants, carbon dioxide, CO2, enters the leaf through the stomata and diffuses into the mesophyll cells and into the stroma of the chloroplast, the site of the Calvin cycle reactions where sugar is synthesized. The reactions are named after Melvin Calvin, the scientist who discovered them, and reference the fact that the reactions function as a cycle. Others call it the Calvin-Benson cycle to include the name of another scientist involved in its discovery, Andrew Benson. Figure 5.14. Light-dependent reactions harness energy from the sun to produce ATP and NADPH. These energy-carrying molecules travel into the stroma where the Calvin cycle reactions take place. The Calvin cycle reactions can be organized into three basic stages: fixation, reduction, and regeneration. In the stroma, in addition to CO2, two other chemicals are present to initiate the Calvin cycle: an enzyme abbreviated Rubisco and the molecule ribulose bisphosphate, RUBP. RUBP has 5 atoms of carbon and a phosphate group on each end. Rubisco catalyzes a reaction between CO2 and RUBP, which forms a 6-carbon compound that is immediately converted into 2 3-carbon molecules. This process is called carbon fixation because CO2 is fixed from its inorganic form into organic molecules. ATP and NADPH use their stored energy to convert the 3-carbon compound, 3-PGA, into another 3-carbon compound called G3P. This type of reaction is called a reduction reaction because it involves the gain of electrons. A reduction is the gain of an electron by an atom or molecule. The molecules of ADP and NAD+, resulting from the reduction reaction, return to the light-dependent reactions to be re-energized. One of the G3P molecules leaves the Calvin cycle to contribute to the formation of the carbohydrate molecule, which is commonly glucose, C6H12O6. Because the carbohydrate molecule has 6 carbon atoms, it takes 6 turns of the Calvin cycle to make one carbohydrate molecule, one for each carbon dioxide molecule fixed. The remaining G3P molecules regenerate RUBP, which enables the system to prepare for the carbon fixation step. ATP is also used in the regeneration of RUBP. Figure 5.15. The Calvin cycle has 3 stages. In stage 1, the enzyme Rubisco incorporates carbon dioxide into an organic molecule. In stage 2, the organic molecule is reduced. In stage 3, RUBP, the molecule that starts the cycle, is regenerated so that the cycle can continue. In summary, it takes 6 turns of the carbon cycle to fix 6 carbon atoms from CO2. These 6 turns require energy input from 12 ATP molecules and 12 NADPH molecules in the reduction step, and 6 ATP molecules in the regeneration step. Evolution connection. Photosynthesis. The shared evolutionary history of all photosynthetic organisms is conspicuous, as the basic process has changed little over eras of time. Even between the giant tropical leaves in the rainforest and tiny cyanobacteria, the processes and components of photosynthesis that use water as an electron donor remain largely the same. Photosystems function to absorb light and use electron transport chains to convert energy. The Calvin cycle reactions assemble carbohydrate molecules with this energy. However, as with all biochemical pathways, a variety of conditions leads to varied adaptations that affect the basic pattern. Photosynthesis in dry climate plants has evolved with adaptations that conserve water. In the harsh, dry heat, every drop of water and precious energy must be used to survive. Two adaptations have evolved in such plants. In one form, a more efficient use of CO2 allows plants to photosynthesize even when CO2 is in short supply, as when the stomata are closed on hot days. The other adaptation performs preliminary reactions of the Calvin cycle at night because opening the stomata at this time conserves water due to cooler temperatures. In addition, this adaptation has allowed plants to carry out low levels of photosynthesis without opening stomata at all, an extreme mechanism to face extremely dry periods. Figure 5.16. Living in the harsh conditions of the desert has led plants like this cactus to evolve variations in reactions outside the Calvin cycle. These variations increase efficiency and help conserve water and energy. Photosynthesis in prokaryotes. The two parts of photosynthesis, the light-dependent reactions and the Calvin cycle, have been described as they take place in chloroplasts. However, prokaryotes, such as cyanobacteria, lack membrane-bound organelles. Prokaryotic photosynthetic autotrophic organisms have infoldings of the plasma membrane for chlorophyll attachment and photosynthesis. It is here that organisms like cyanobacteria can carry out photosynthesis. Figure 5.17. A photosynthetic prokaryote has infolded regions of the plasma membrane that function like thylakoids. Although these are not contained in an organelle, such as a chloroplast, all of the necessary components are present to carry out photosynthesis. The energy flow. Living things access energy by breaking down carbohydrate molecules. However, if plants make carbohydrate molecules, why would they need to break them down? Carbohydrates are storage molecules for energy in all living things. Although energy can be stored in molecules like ATP, carbohydrates are much more stable and efficient reservoirs for chemical energy. Photosynthetic organisms can also carry out the reactions of respiration to harvest the energy that they have stored in carbohydrates. For example, plants have mitochondria in addition to chloroplasts. You may have noticed that the overall reaction for photosynthesis, 6CO2 plus 6H2O produces C6H12O6 plus 6O2, is the reverse of the overall reaction for cellular respiration. 6O2 plus C6H12O6 produces 6CO2 plus 6H2O. Photosynthesis produces oxygen as a byproduct, and respiration produces carbon dioxide as a byproduct. In nature, there is no such thing as waste. Every single atom of matter is conserved, recycling indefinitely. Substances change form or move from one type of molecule to another, but never disappear. CO2 is no more a form of waste produced by respiration than oxygen is a waste product of photosynthesis. Both are byproducts of reactions that move on to other reactions. Photosynthesis absorbs energy to build carbohydrates in chloroplasts, and aerobic cellular respiration releases energy by using oxygen to break down carbohydrates. Both organelles use electron transport chains to generate the energy necessary to drive other reactions. Photosynthesis and cellular respiration function in a biological cycle, allowing organisms to access life-sustaining energy that originates millions of miles away in a star. Figure 5.18. In the carbon cycle, the reactions of photosynthesis and cellular respiration share reciprocal reactants and products.